Hi guys, it's Audrey, Claudia, and Charlie, and we're here to talk about our IB biology unit 10.1 through 3. This is our project. So, if sperm donors are more commonly used, how will the gene pool be impacted? Sperm donors and genetics. Sperm donors are becoming increasingly popular as a number of lesbian couples, women choosing to stay single or have kids, and infertility issues arise. Here's an example of a few sperm donor families. I think you can read these for yourselves, but these are some of the
Horror stories.
yes, these are accidents, or this is also what could happen with sperm donors. Like there are many beautiful things, but there are also less beautiful things. So, some vocabulary that we're going to be using throughout this presentation: haploid, when a cell has a single set of chromosomes; diploid, when a cell has two sets of chromosomes. A chromosome is a strand of nucleic acids that codes for genetic information. A gamete is a sex cell. And mitosis is the process that gametes go through so they can replicate.
So, meiosis is the process by which gametes are produced, and gametes are, like Audrey said, sex cells, so sperm and egg. It's similar to the process of mitosis, but it happens twice. So, instead of creating two diploid cells, meiosis produces two different haploid cells, which are all genetically different from each other. Oh, four, sorry. and Meiosis is also random, which also, along with the genetic difference between the four haploid cells, helps to provide genetic variation that helps drive evolution. So, like I said, there are two, meiosis happens twice. So, there's meiosis one and meiosis two. But before you can get to meiosis one, you have to go through interphase, which is when the DNA replicates so that all chromosomes consist of two sister chromatids. And so, meiosis one of the most important phases in meiosis one is prophase one. And this is when the chromatin created in interphase condenses into chromosomes. And these replicated chromosomes pair up into a tetrad. And then metaphase is when the tetrad moves to the middle of the cell. Um, and anaphase is one of another very important phase in meiosis one. This is when the homologous pairs, which is what the tetrad is made up of, this is when they're pulled apart to opposite ends of the cell. And then telophase is when two new cells form. And this first cell division is what results in two haploid cells. And so, next, of course, is meiosis 2. And meiosis 2 starts similarly um, to meiosis 1. The chromatin condenses into chromosomes, and since they're already replicated, they're not homologous pairs. Metaphase 2, just like metaphase 1, the chromosomes are pulled into the middle of the cell, and anaphase 2 is another very important phase in meiosis because the replicated chromosomes at this point are pulled apart and pulled to opposite ends of the cell. And then telophase 2, so the two haploid cells that are produced in meiosis 1 both divide. And so now you're left with four haploid cells that are all genetically different, and these are called daughter cells. So these are just some images of the stages of meiosis, just to give a visual representation of the most important stages. So, like I mentioned earlier, genetic variation is one um, product of meiosis, and that happens through the random nature of meiosis along with other factors. And so, one of the things that causes genetic variation in meiosis is crossing over. So, this happens in prophase one, and this is also what makes prophase one of the most important phases in meiosis. Um, so, this is when the two non-sister chromatids cross over because in the tetrad, because they're sitting so close together, sometimes their alleles mix up. Um, and the spot that at which this happens is called the chiasma. And so another thing that causes genetic variation is are mutations. Mutations happen randomly and they can be either beneficial, detrimental, or they can not cause anything. They can just be there. Um, yes. And Gregor Mendel, one of the first people to observe genetics, he had um, three different laws that help describe meiosis and its genetic variation. So first is a law of dominance, which deals with the dominant and recessive traits that show up in on chromosomes, like with our alleles and things like that. And this law basically states that the dominant gene will always show over the recessive gene. Next is the law of segregation. And this just states that the homologous pairs that are seen in meiosis 1 separate randomly. There's no rhyme or reason to it, which is what contributes to genetic variation. And then the law of independent assortment, which once again is random. And this is when the gametes join. They don't like look for each other. It's just a random process that happens and contributes to genetic variation.
Okay, so next is inheritance. Inheritance refers to traits encoded in DNA that are passed from parent to offspring in reproduction. Typical traits and mutations can be passed to the next generation. So, how does inheritance apply to our project? Since mutations are very common, sperm donors are screened for serious diseases. However, many doctors recommend genetic matching to ensure that the risk of genetic disease is as low as possible. Genetic matching screens both the parent and the potential donor to prevent combinations of two recessive alleles or two potential carriers. This produces a lot of risk, especially if the donor doesn't disclose all potential information about what diseases they have, and they might not even know that it's a genetically caused disease. So this that could cause some problems for a child in the future. So here's one example. Um, so a sperm donor father donates to multiple mothers. Two of the kids unknowingly end up getting married and having a child. So how would that affect the child's genetics? Well, so there's two Punnett squares here. And so the top two, or the top of the Punnett square represents the sperm donor, and then the two side columns represent the mothers. So mother one is a homozygous recessive carrier, and we're gonna call C curly hair. And then mother two, uh, big C is curly hair because it has, because you use the dominant trait based to determine the letter. And so then mother two is heterozygous. So the Punnett square showed the potential for their children. And so this example is using autosomal chromosomes, which are just non-sex cells. Um, so same situation. The potential genotypes for the kids. So the first kid could be 50-50 dominant or heterozygous for curly hair or recessive or homozygous recessive. And then the second child could be uh, 25% dominant homozygous, 50% heterozygous, or 25% recessive homozygous. So, yeah, just clarifying again that this is a that curly hair would be the dominant trait. So continuing with the problem, the child's genotypes, if we're looking at it, if we're looking at these two, since child one was 50-50, we just kind of randomly chose one of the potential things. And then since child two had a higher percent of being heterozygous, then we chose to use that one. So this would show, this Punnett square shows what their children would look like. So their child would potentially have uh, heterozygous or homozygous dominant 25%, or heterozygous 50%, or heterozygous homozygous recessive for 25%. And so that means their phenotype is 75% curly hair and 25% straight hair. So they're more likely to have curly hair. This is like starting to limit the gene pool, which we'll talk about in the next section. So, this child, yeah, so that's what I already talked about. But what if a sperm donor has an undisclosed genetic disorder, like we talked about a few minutes ago? How could the child be impacted genetically? So, this example is going to use color blindness, and N is chosen as a allele because it's the normal condition for color blindness. So the mother this time is on the top and the sperm donor is on the side. The sperm donor obviously is male and the mother is obviously female. And so this is also showing how a uh, cross with a sex linked cross works. So you have to both do the sex determining and the trait determining. So the trait is usually represented as a superscript letter, like almost like an exponent in math. And so, using this example, the potential children are a female carrier, which just means that they potentially have, that they're just a carrier like the mother is. And so they just have the recessive trait. They don't necessarily have the condition. Or it could be a colorblind female or a normal male or a colorblind male. So these are the different. So there's a 50% chance of the child being colorblind, which isn't that bad because it's just colorblindness. But this example also can apply to much more serious sex related traits.
So this impacts gene pools, as Claudia already said, but it can also, not only does it affect the gene pool, but it can really, really hurt the people because of that. So, and this is because of a kind of accident or mistake or just a like a little oopsie by the sperm banks, because many sperm banks sell hundreds and hundreds of units from the same donor to different families, like, and they don't regulate it, they don't track where the babies go or if it was successful. So now in theory you could have 100 babies created from the same father, which creates a smaller gene pool, and then that situation with the incest that can happen again. So allele frequency, this is also affected by genetics or the gene pool. So it helps us to understand how evolution is going and what happened in the past. So it's presented as a proportion or percentage, and we'll talk about that more in the next slide. Eukaryotic organism alleles occur in pairs, known as genotypes, and we already talked about that. Interbreeding populations. In most populations, the gene pool is larger and there's a large variety of traits present, but with interbreeding populations, there's low variety. So the Hardy-Weinberg equation shows how we talk about allele frequency. This equation is used to calculate the frequency of genotypes and phenotypes. And it can also predict the outcomes of certain crosses and determine the rate at which a population is changing, which can be really, really useful. just for genetic studies or the course and seeing about this exact study, how what's going to happen if we keep using sperm donors.
So the use of sperm donors has a lot of genetic implications, but there are also some ethical considerations that need to be addressed when talking about the use of sperm donors. So one ethical issue with sperm donation is that many sperm banks will only accept donations from certain types of people. So, men of a certain age or IQ, or even physical features. One sperm bank, which is also happens to be uses a dating site, uses this slogan, "Who doesn't love a pretty baby?" Which a lot of people could argue that this is kind of eugenics, because there are certain people that a lot of sperm banks just will refuse to donate sperm.
But I mean, in theory, you could make the same claim about a woman having sex with a pretty man to have a pretty baby. But it's all a strange conversation, that's for sure.
So there are a lot of different rights involved. Of course, every person involved in the process of sperm donation has different rights and obligations. So here are a few. From about the client, the person who's receiving the sperm, and the person who's donating. So for the client, they have the right to be informed of any limitations and complications with sperm donation. Um, because it's not a foolproof system, things happen, and they have the right to be informed of that. Um, they also must understand that genetic testing is not, as I said with the complications, is not a foolproof system. And so the sperm bank cannot 100% guarantee that the sperm that they use with the client is 100% without genetic mutations. And that's one of the major ethical issues, because you could be giving someone sperm that has a really rare, really harmful genetic disease, and now they have to they have all of this unprecedented responsibilities and problems with their child. Um, the client is also fully responsible for the offspring that's produced from the sperm that they're given. The donor is not considered the father, they're the sperm donor. Um, and also, they have no right to try and find the identity of the donor, or to solicit that information from someone. And the donor, their identity may remain anonymous. That's really up to them. And the donor has no obligation, like I said, to their biological offspring. They're not considered by the sperm banks, or in most situations, the court of law, they're usually not considered the father.
And these are our sites that we used. Thank you for listening.